Copyright Stanford University, all rights reserved.
Good morning. My name is Kim Butts-Pauley, and I'm the director of the Center for Biomedical Imaging at Stanford. And the goals of CBIS are to bring together the Stanford community and to facilitate research involving imaging across campus. And I'd like to draw your attention to our annual symposium, which will be held here at April 5th and 6th, and it'll be done in conjunction with the Stanford Center for Image Systems Engineering. It'll be in this room. We'll have Dr. William Brody from the Salk Institute, and we'll have opportunity for Stanford speakers and posters, and it'll be a great event. So I'd like to draw your attention to that. Today, we're delighted to have Dr. Elias Zerhouni here for a special seminar. I first met Dr. Zerhouni about 20 years ago at a meeting on magnetic resonance imaging and CT, and at that time, Dr. Zerhouni was director of the MRI division of radiology at Johns Hopkins University. He did a lot of great work in cardiovascular MRI and was a developer of myocardial tagging. He was extremely creative and a real pioneer of new techniques in MR and CT. Dr. Zerhouni went on to serve as the Executive Vice Dean of Johns Hopkins University School of Medicine from 1996 to 2002. He founded 5 startup companies, including Computerized Image Reference Systems, Advanced Medical Imaging, Biopsis Corporation, American Radiology Services, and Surgivision, which I believe has a new name now. He's authored over 200 peer-reviewed publications and holds 8 patents. In 1985, he was consultant to the White House, and in 1988, he was a consultant to the World Health Organization. Since 2000, he has been a member of the National Academy of Sciences Institute of Medicine. He has served on the National Cancer Institute's Board of Scientific Advisors from 1998 to 2002. And as we all know, from 2002 to 2008, he served as the director of the NIH. While at the NIH, Dr. Zerhouni promoted global health and global research, and since leaving the NIH, Dr. Zerhouni moved to Sanofi, a company dedicated to global health care. He is currently the president of Global Research and Development and a member of the executive committee for Sanofi. In addition, he is a senior fellow of the Bill and Melinda Gates Foundation. He is on the board of trustees at the Mayo Clinic and the Lasker Foundation. He is a member of the Institute of Medicine and the U.S. National Academy of Sciences. He received the prestigious Legion of Honor Medal from the French National Order in 2008. He was recently appointed as the chair of innovation at the College of France and elected as a member of the French Academy of Medicine. So we're really very honored today to have him speak to us about the challenges facing biomedical research and development. So please join me in welcoming Dr. Zerhouni.
Well, thank you for this very kind introduction. And as I say, great introductions like this are something you should record and give to your mother-in-law to play. This is always my... But the other thing is I'm honored to be here because I have a long-standing attachment to Stanford Radiology. I know Dr. Abrams, I know Bill Northway, and I had colleagues here, but most importantly, I was close to Gary Glazer when, at the time, he became chairman. I was connected with him at the University of Michigan. We did a lot of work together in MRI in the early days. And I've always followed Gary's career, and we've always been colleagues at a distance and friends. And I wanted to come because I couldn't come for the memorial. And so I told Sam, I said, the first opportunity I'm here on the West Coast, I'd like to give not only this lecture, which Hans Ringers had been begging me to come and find a time to come to Stanford. So first opportunity I came, but I really want to dedicate this lecture to Gary, and his leadership and what he's done for this department is remarkable. Really, from what I remember was Stanford Radiology in 1988 to what Stanford Radiology is today. So I'm very honored to be here and share that with you. What I'd like to do today is not give you an imaging talk. I'm going to weave imaging concepts in there because I think imaging has a huge role to play in modern biomedical research. But I'm going to give you my worldview about what are the trends, what are the challenges, what is really in front of us as both physicians and scientists, what we can see as ways of approaching some of the issues we're facing. So first and foremost, I'd like to just give you a quick sort of synopsis of the past 100 years. And there's no doubt that biomedical research has led to great progress, especially in the control of infectious disease and acute diseases. Today, most of healthcare spending is spent on chronic diseases. 80% of all the healthcare budgets you hear about that are really bankrupting governments around the world are related to chronic diseases, not acute diseases. In the meantime, over the past 50 years, I think you can characterize the period of history, of scientific history, as one of rapid discoveries of the fundamental elements of biology. And essentially think of it as a period of a reductionist exploration where the questions that were posed in the 40s were answered between 1950 with the elucidation of the structure of DNA all the way to today, where the research has been, let's identify every fundamental component of biological systems. It would be DNA, RNA, proteins, their role, their interactions. And that was a splendid period of history. It's still ongoing. But I think what has happened over the past 10, 15 years has been the ability of scientists to put together these fundamental elements in what I call modules. So modules, for example, functional modules that describe components that relate to replication or transcription, translation, all of the fundamental elements now of an integrated network of molecules that do achieve some functions. And also because of genetics, our understanding of the evolution of the systems, of these subsystems, these modules, has really become greater over time. We do know how to understand how a system has been evolving over 2-3 billion years and how it's translated in different species. And so I think the research tools that have emerged have really allowed us to understand biology in a different way and really allow us to see a new period where integration of all the knowledge we've accumulated is now possible, at least at the fundamental level. In the meantime, if you look at public health, and now I'm coming back as a physician, as a public health person, what are the challenges in front of us? I'm not going to surprise you by telling you that the number one is this shift from acute to chronic conditions, which we haven't really been able to overcome at this point. Aging of the population, and it's the paradox of medicine. The more successful you are in medicine, in prolonging lifespans, the more the demand will increase because you basically have a larger population asking for more care. Now, global health disparities remain an issue. I think the world has seen a huge demographic expansion. I think we've seen wealth expand in the world with more people coming out of poverty than ever in the history of mankind. But at the same time, because of fast demographic growth, in absolute numbers, the number of poor people below $1 a day has increased. Even though in relative numbers, we've seen progress, especially in the middle class, in China, Asia, India, and Brazil, for example. So it's interesting because at the same time, you're seeing great progress economically because of the demographic expansion, you're also seeing in absolute number a greater challenge in the absolute number of people who really need us to find solutions to global health problems. Emerging and re-emerging infectious diseases due to, again, a change in urbanization, transportation, demographics, and also, as you know, the emergence of non-communicable diseases like depression, which is predicted to be, if not already, by the WHO, the #1 cause of disability in the world between the ages of 25 and 44. A huge burden in terms of unipolar depression, allergy, the rise of asthma, for example, and obesity with its attendant consequences, diabetes. And obviously type 2 diabetes is only the entry door to a Pandora's box of chronic conditions and chronic comorbidities like heart disease and renal disease and eye disease, nerve disease, and infectious disease. So much so that this conglomeration of pathologies around the concept of obesity and diabetes is becoming now called diabesity, and frankly is probably one of the biggest threats to public health as we are facing it today. So as we go forward, the question that we have to ask is there a change in paradigm that is going to be necessary or needed or welcome or not? Do we want to think of what you will call a change in our operating model in medicine. And clearly, I think it's obvious that we are already ongoing that change of paradigm. And how do I describe this? The best way I can describe it is that if you really look at any disease burden, you can always divide the phenomenon of disease development into a preclinical phase, a sort of compensated phase, or an uncompensated phase. So these are the three phases. And over time, obviously, a patient traverses the preclinical to the compensated and compensated phase. It's a very classical scheme. The problem is that costs do not also evolve linearly with degrees of complexity in the disease process. They actually evolve exponentially. So if you are, for example, a patient with a little bit of elevation of blood pressure, and you're in the early preclinical phase, you can probably take care of that with diuretics. It's 50 cents a day. And then if you go a little bit further and you have high cholesterol, and you probably need aggressive primary prevention methods, and it's probably $5, $10 a day. And then if you go to the compensated phase where a stress test identifies problems or a cardiac scan score or anything like this, and then all of a sudden you're talking about managing this disease with significant medication, and that's $100. I mean, $10 a day, 1 to 10. And then if you have a real angina, it's $10 a day. And then if you have symptoms, you have to get a cardiac surgery and for a bypass, and you're talking about $100,000. And then if it becomes uncompensated, then you have a heart transplant, it's $1 million. So what I'm trying to tell you is that there's a logarithmic growth in costs as you intervene later and later. And that's why the economic model cannot be sustained at the curative treatment phase, where we just wait for things to happen at the decompensated phase. And that's the tradition of medicine. I mean, you don't see the intervention until very late in the disease process, when pain occurs or loss of function occurs. And that's the paradigm of the past 5,000 years. That cannot be sustained, and over the past 30 years, we've seen the evolution of what we call symptom management, where in fact, as soon as the disease gets into the compensated but obvious clinical phase, you intervene with statins or other interventions, lower blood pressure, and that has made a huge impact. So symptom management approaches have been essential to the control of acute conditions like heart attack, stroke, and so on, with a 70% drop in mortality for cardiovascular, coronary heart disease, and stroke. Really remarkable. However, we also know that this is not going to be sufficient. And clearly, we will need to intervene much earlier and preempt the disease. In other words, a chronic disease does not occur overnight. It occurs over 20 years. If you look at many of the conditions we're dealing with today as researchers, I mean, diabetes, you can see evidence of that 20 years before it becomes clinically obvious. Alzheimer's disease. And I think so the whole efforts of the past 10, 15 years has been, can we characterize patient populations and stratify risk within patient populations so we can identify those who are more likely to end up in the phase of decompensation from which we cannot recover. And so the whole idea percolating in the research world over the past 10 years has been, can we, through genomics, pharmacogenomics, other methods, identify these categories of patients? So we funded studies, for example, in Alzheimer's disease, imaging of Alzheimer's disease, to try to find markers or biomarkers of disease. And the reason you want to do this is twofold. One is, epidemiologically, we know that the number of patients to whom we give statins is much larger than statistically, the number of patients who would eventually suffer from a coronary vascular event. So if you look at, if we give it to 100 people, statistically only 10 to 15 may ever suffer from that. So we know that we're giving it to 85 people, and the outcome of that intervention is just there's no evidence for it. The only problem is we don't know which 15 to give it to and which 85 not to give it to. So that's the point that we're trying to make. Alzheimer's disease, the same thing. There is no way that we can develop a treatment for Alzheimer's disease not knowing of all the patients who may have temporary loss of memory when you don't know where your keys are, whether or not it's a precursor of Alzheimer's disease or not. You need better markers for that. But the most important reason for that stratification is because we do understand that the fundamental molecular mechanisms of diseases that appear as one disease are not uniform. In other words, if you look at phenotype, there are many genotypes that are the pathway to the development of a disease process. So when we talk about pulmonary fibrosis, I see Bill Northway is my friend from the Fleischner Society. I mean, we know that pulmonary fibrosis is not one disease. Breast cancer is not one disease, and so on. So molecular subclassification of disease is ongoing today. So we are trying to understand at the population level, what are the risk factors that are predictive? And at the same time, we realize that to do that, we need to understand the molecular pathways of disease much better than we do today, which really opens a completely new trend, and that is an effort at complete reclassification of our disease states. And that reclassification is eventually going to lead to what I call a molecular preemption capabilities, where you could detect the disease early and intervene at a stage where it's still compensated rather than decompensated. So that's the whole strategy, if you will, that all of us hope for and work towards. So if you said, all right, how can you summarize this? Basically, this slide tries to capture that. In the 20th century, we treated disease when symptoms appeared, you went to the hospital, and normal function was lost or pain was evident. We didn't really understand the molecular and cellular events that lead to disease, even though today we don't understand this fully as well, but we do better. And the ability for us to take care of a disease state like this led to a fragmented system and expensive and financial and disability costs, not just financial care costs, but the impact on the patient. So it's clear that we're going to have to intervene before symptoms appear and preserve normal function. And that is because we need to make a greater effort at understanding preclinical molecular events, and our ability to detect patients at risk is going to be paramount. And I say preclinical molecular events. And that means studying these disease patterns in patients. And then the focus obviously has to be on these growing unmet needs. So this is what I call the future paradigm, the 4 Ps, you've heard this. And basically the 4 Ps means predictive medicine that is personalized, that is preemptive, but will require participation. And the participation is required because we're not going to have patients who suffer from a disease. They'll have to make the proactive move of being screened or have a genome scan or a diagnostic scan so that we can really provide precision medicine. That's the overall scheme, if you will, summarized. Now, when you look at where we are we doing well? And the answer is no. The answer is neither academia, nor government, nor industry is doing well in trying to understand and figuring out what it is that we need to do and getting treatments for patients. And the best thing you can see is something that was quite obvious to me when I was at the NIH. I used to meet with the heads of R&D from all the world, and what was obvious is that there was a huge rise in R&D expenditures. So if you look at the NIH budget, it's about $30 billion a year. And if you look at total R&D expenses in industry, it's $65 billion. So it's twice what the government spends. And then when you look at the number of new molecular entities approved, it went down from the 90s. And there are many reasons for this, but the key thing is that we have a productivity crisis in science. We have great understandings, better tools, better models in the laboratory, but they're not being translated into obvious, realistic, effective cures for millions of patients, especially in what I call the primary care threats: obesity, diabetes, degenerative diseases. So we're not making the progress we need to make. And that's an issue that I feel very close to, and frankly, I think we should be accountable to it, because the public has been funded has been funding scientific research and biomedical research at great expense, and there's a high level of expectation for solutions. And we've been sort of creating these expectations. Now, at the same time, we've made enough progress, in my view, that we can start thinking about a different era where we're going to perform R&D differently. So if you really think about it and you say, well, what's the problem? The problem is not organizational. People think it's organizational, it's healthcare systems, insurance companies. I happen to believe that the fundamental problem is scientific. And the problem is that our ability to study complex and dynamic biological systems in health and disease is very limited. And yet, today, the major diseases that affect us are really complex diseases with multiple, like I said, multiple molecular pathways to their phenotype. So think of it this way. If I had a way to cartoon this, think of biology as a multi-layered, complex network, which has its own layer. Each layer itself has its own regulation, but interacts with the next layer. What do I mean by this? So let's say this is the DNA, and let me simplify by every color here. It's an exon, for example, that codes for a protein of the same color. Well, we know that, transcription and translation occur, but these proteins are not independent. They're really interconnected in networks that have functional interactions and feedback loops. And just to make it simple, an arrow means it promotes the activity of C, and a T, is a repression. For those of you who know these parameters, it's clear that this is sort of the mental set that we've had in the past 20, 25 years as scientists, saying, well, if I find a DNA error, if I find something in the DNA that's a misspelling, like a polymorphism that leads to an allele that is dysfunctional, well, I could probably trace that down the chain of biological events. So it's the DNA, RNA, protein action theory, which has been the dominant one. And clearly, what we're discovering is that that's not the case. It's a little more complex than that. So if you had, for example, a DNA error, you'd have a malfunctioning protein C, and this protein C, therefore, will no longer repress A, and it will no longer promote D, so that when you observe that at the protein level, so if you did a blood test and you measure the protein, what you would see is that A will go up, E will go up, and D might go down because it's not, but unless you understand all these changes, it will be hard for you to detect the fact that this is the cause of the disease, not the fact that, cholesterol goes up or something else goes down. So these networks are not linearly or collinearly expressing their function. And therefore, when you look at a disease state, you really need tools to better understand the complexity of this network, not just in one snapshot, but dynamically over time. And if you then say, well, that's good. Well, at least if I could do this for this particular abnormality, I'll be doing well. But let me give you another example where, in fact, the DNA abnormality is in B, and B basically leads to a hypoproduction of protein B, which then leads to hypoproduction of C, which then prevents the repression of A and prevents the overcreation of D. So if you look at this, well, A is overexpressed and E as well, just like we had it in the previous scan, in the previous case, right? There are some slight differences. B here, for example, is a little bit more than it is in the next case, just because of the mistake C. But unless you can measure all of the actors and their response, it's impossible for you to realize that you had the same, what looked like the same disease, with A being out of whack and E being out of whack, when in fact you didn't have the same disease, you had two. You had one in C and one in B. So what I'm trying to say here is that we have a fundamental issue in front of us, and that is one phenotype can be under the influence of multiple genotype. And so when you think about it and you say, okay, what's the biology that we need to comprehend to be able to truly be effective in discovery, you realize that you have to understand the biology. It's almost like an onion peel. You have multiple layers, Obviously, epigenetics that controls DNA, the genetics and genomic codes, the next step and regulation as well of all of these, and proteomics and metabolomics and all the omics you can think of. But all of them are not necessarily connected in ways that are predictable unless you can truly study at high resolution, both temporal and quantitative, the changes in the different elements. And we're getting there, and it presents a problem because the amount of data needed to understand even a simple module is enormous. So reverse, the inverse function of what you observe into understanding the cause of disease is quite complex, quite difficult. And that does not take into account the environment, because I think people always forget that whatever functional set is present is actually driven a lot by your environment, which can affect both epigenetics and many other of the components of the system. And so environment, gene interaction become critical. And if you really ask yourself, how much do we understand of that in diabetes? Well, I will tell you, less than 10%, if at all. There are many things that we do not understand in diabetes. When you ask the question, there's a lot of hand-waving, but frankly, there isn't a lot of what I would call solid evidence in the interactions of environment and the biology. Now, I like to show this slide, and I would like to say that one of the great things about being an NIH director is you can get some grants done, some research done without going through the peer review process. But it has to be less than $50,000. So I was able to do some studies and experiments that no one else could do. So one of them is I asked the guys in Florence to send us Michelangelo's David statue, because I said we need to do an experiment that separates environment from genetics. And I knew statues had no DNA. So we knew that if anything happened, it would be an environmental factor-driven thing. So we brought the statue to the US for about a year. And that's what happened. So clearly, so that was a very short, small, sweet grant to prove that environment is important. Anyway, so to reveal my biomedical engineer background and my way of thinking, I'm showing you here what I think is the scientific challenge in front of us today, and how to tackle this biological complexity by showing an electronic diagram. It'd be like a chip that you would get from a computer today. And suppose your task is to figure out what the chip in the Intel inside chip is really doing. Well, the first thing you would do, you try to evaluate the elements of the chip and the transistors and so on. But you could do that for years and years and years and years. It will never tell you how a PowerPoint presentation is made, how a WordPerfect document is made. It's the same thing in biology. I mean, you can look at a cell response element to damage. You can see all the elements, all the cytokines, and all the signaling and the T cells that interact and all the proteins underneath. But it doesn't give you the software. And I think this is where we're facing right now a challenge in front of us, and that is that we have become very good at understanding the components, and I think we need to become good at understanding what I call network biology, quantitative network biology. So that's my fundamental vision in trying to say we will not be able to make progress without a true effort on understanding, at least in some modules, some of our fundamental inflammation, immunology, degeneration, something that relates to pathology that we can identify as a module that we can completely map out and quantitatively understand in terms of the network that interacts to define the biology or its the pathology. So if you take one thing from this lecture, I believe we're at an inflection point where we're going in a phase of research that was driven by the hardware of life, if you will, to the software. And that means understanding molecular pathways and the regulation in health and disease. This is something that when I was at the NIH, I launched the roadmap for medical research. About 40% of our efforts were driven to that. The second is that it will lead to a functional and more precise reclassification of most diseases based on their specific molecular pathways, and obviously better understand environmental drivers. So that's the posit that I'm bringing you to. But here's a more important observation. One of the reasons I think we've not been as successful in biomedical research at all levels in translating is because a lot of the models that we use in the labs are not predictive of what happens in the human body. And so all of this work has to also be done in the translational context. That's why I was a big champion of translational medicine approaches in the sense of bringing our methods, the tools that we develop in basic research at the bench to really understand disease in the human context. And that requires an enormous amount of innovation. A lot of it will come from the imaging community. A lot of it will come from the imaging community. For example, today, as you know, we have personalized cancer care. And we do understand some of the driver mutations. We do understand some of the pathways. The thing we cannot do is measure comprehensively in a single patient what is the molecular response of the patient to a particular treatment. We can't do this. And frankly, I know Sam is really a big leader there in terms of molecular imaging. It's not sufficient. We're going to have to develop techniques that I can't even think of, but that will probably relate to proteomics and sequencing and other things that will allow us to have a window on the pathology at the time we are trying to test out these hypotheses. Subclassification of disease, it's going to be very, very critical to progress. So I'm just going to go forward by saying that, okay, if you agree with me, and if you say, I think you're right, we need quantitative network biology, and we need to get to the software. That has implications on the way you conduct biological experiments. It has profound impact on the kind of data that you will need to generate to even be able to approach the question, not solve it, approach it. And what does that mean? One, if you're testing a system and you want to read it over time, it cannot be a destructive method. It has to be a non-destructive method. So we are going to be going from destructive analytical methods to non-destructive. And there are many of them that are really coming up that are, I think, very promising. From qualitative to quantitative. It's not enough to know that a gene is up or a gene is down. By how much? What else is down? What else is up? We need to have this quantitative relationship. Can't interrogate, as I showed you, one parameter at a time. You're going to have to really do this in multiple dimensions. The other important phenomenon is that we now know that even localization of molecular events is very critical. For example, in cystic fibrosis, we know the mutation. We know, since 1988, we've known the gene. But the gene itself, the protein in vitro works. It's functional. The only problem is that it doesn't get to the right place. It stays in the Golgi in the cell because it doesn't have the addressing mechanism to bring it to the cell surface to function as a channel. So here's an example where observing the localization was essential to our understanding. So it has to be temporally resolved, spatially localized. Anyway, I don't want to belabor the point. I think you understand that in a high data density world, we won't be able to extract scientific truth without common standards and sharing of the experiments. So to me, if you say non-destructive and quantitative, multidimensional, high temporal resolution, spatially resolved, high data density, common standard, our own DICOM standards, and the ability to accumulate that, I think you have a sort of a back-ended definition of imaging. And that's why I say imaging will be key. So now I'm going to tell you about what my view is about the role of imaging in this sort of biomedical revolution that needs to happen, or is happening, actually. So I'd like to go back then, and in the second part of my lecture is to connect imaging to what I just said. And, I see a lot of you here have trained with me. I see Rusty over there. And we were trying to say, okay, what's the essence of imaging when we were doing that at Hopkins? And fundamentally, our analysis is this, is that no imaging can be created without a discovery of a basic physical process of imaging tissues. So X-rays, there's absorption by atomic nuclear layers, CAT scanning with a computer, and so on and so forth. So there's a fundamental basic process that can capture information from tissues. Then the second is that as soon as you have that, naturally you want to display anatomy and structure. And that's the natural history. So the hand of Röntgen's wife with the ring, I mean, that's anatomy and structure. But very, very soon after that, the use of biological knowledge helps you enhance the results of structural results, structural imaging results. So for example, you learn about swallowing barium so you can see the stomach. Well, that's a contrast agent. And then you hear about biliary metabolism, and then you create the first oral contrast agent for the gallbladder. And then Igasmonides says, why not inject that in vessels? And he creates angiography. So all of a sudden, you expand the field of the imaging method. And that story, by the way, you think it's true to X-ray? It's true to every method that has come except probably PET, because PET didn't have the temporal resolution. But MRI, we went from anatomy, structure, vessels, and other functional measures, because we knew the biological method, I mean the phenomenon, and we used this to study function. So it goes from anatomy to structure to function. And then what typically happens is that you have progress in other key technologies. And this is where imaging really, to me, is the quintessential example of a multidisciplinary research field. You cannot make progress in imaging unless you combine, in fact, all of the sciences needed: physics, image processing, computing. And this is exactly what has happened to the world of radiology. I think we've been able to combine different things as they came along. So TV, for example, came along, and I remember Russ Morgan, who was the chair of radiology at Hopkins, is also an engineer, and he first and foremost combined an oscilloscope with a TV. And that opened up the whole field of interventional radiology. And with Dick Ross, he did the first interventional procedures. Dr. Abrams is here to tell you about how the initial research in Sweden, as well as in the US, created the whole field of angiography, and it's splendor because you could do it. You could interact with the system. So we go from static images, functional images, interactive, because we use different technologies. So real-time cines, computer technology then came along, and that just exploded the field, because all of a sudden, you were no longer just limited by the physical process, but you could really exploit the physical process to all its dimensions, especially in MRI, for example. And then the cycle closes, because then new biology it sort of drives new technology. So I was just talking to Sam. We needed a biomarker to evaluate cancer in particular reasons. And so we came to him because that biology can be, in fact, elucidated by what he's developed as a technology. So essentially, I think we are in the computer imaging era. So from my standpoint, for the students here, the way I see it is that you have an energy field that interacts with the object, is detected, and now we have a computer that basically just creates an inverse function, if you will. It transforms the data into something that you can observe, but something also you can compute. And pretty soon, as you will see, there are images that we will no longer observe by the human eye, but they'll be observed and interpreted by segmentation. So the innovation cycle is physics, computer software, image formation, biology, and back to image formation, back to imaging physics again. So it's a cycle of innovation that goes on. So how are we going to, how's it been? Clearly, if you ask physicians in practice and you ask them what are the top medical advances in the past 30 years, and it's clearly in the past 30 years, there are significant advances that have impacted medicine. MRI and CT is always cited as tops. Obviously drugs, ACE inhibitors, but balloon angioplasty, mammography, coronary artery bypass graft, hip and knee, all of which are dependent on good imaging. And I think the yin-yang between biology and technology is really the message here. As Shirley Tillman says, new ideas are great, but you can't really advance them without technological progress. So it's a combination that creates a virtuous cycle there. So the first observation I'd like to share with you is that imaging will be relevant at all scales. It's not just organ imaging, structural imaging. And that means that from microns to meters, you need to think about all the applications of imaging. And it's really happening as we speak. I mean, you'd be amazed to see all the experiments that have been made possible by the discovery of GFP protein, which are fluorescent protein at the microscopic level. And photoactivated light microscopy today, I mean, you can see molecule by molecule in the mitochondrium what is really happening during the cell cycle. So these things are going to be true at all levels. I don't want to belabor the point. I just brought in some of my own research in the old days where we were going into dynamic imaging for MRI. I see Scott Reeder here. He may remember some of that work. But here in this case is a woman that had a multiple emboli to the lower extremities. And we saw this defect here. And obviously with image processing, no, don't do this to me. Oops. I don't know what's happening. This is the PC to Apple transition problem. Maybe this time. So anyway, what I was just going to show you is that, you know, internal navigations, this is old stuff. I mean, this is like 15, 20 years old. But it just showed you that you could envision using these methods from large-scale imaging to micro-scale imaging. And Ron Germain, and again, the movie doesn't work. This is actually a lymph node, and the green cells, the green label cells are circulating neutrophils in a lymph node, and it is fascinating what happens. I'm sorry, the movie doesn't work, but basically understanding what happens in a lymph node with what cells approach which cell and transfer what molecules and the signaling of that ballet of what happens in the lymph node is so revealing of what the immune system does that this has become sort of the mainstay now of a major branch of immunology, what we call dynamic imaging of immune responses. This is a famous movie. This is a macrophage tracking a bacterium right here. So I'm playing it again. This one, we're lucky it plays. So this is the macrophage turning and chasing the bacterium here. You see a nose cone. Now here's like a radar that it tracks the bacterium. You know, the bacterium tries, I don't know if the bacterium tries to get away or is pushed. I have no idea, but it's really being chased. And you see the dynamic behavior of that cell. So I ask the students always, I say, well, you know, this little movie here, in my mind, is really posing 30 questions. This is the beauty of imaging, because how does that cone form? How does the detection cone? I mean, this is almost like a cruise missile, you know, with a radar in the front and going after the target and chasing it. How does that cone form? What proteins are there? What makes the cell be able to sense the bacterium? What is the signal and the physical, you know, the chemical gradient? How does it even know how to move right or left? I mean, there's no legs and you can imagine whole fields of cell biology being obviously there and obviously being stimulated. And you can have an assay that basically says, can I stop this macrophage from chasing the bacterium? Well, you could. I mean, you could create an assay, and we could get a full library, and oh, gee, we would find probably compounds that will stop the macrophage from going after the bacterium. And how good would that be? Well, maybe if I can do this with a T cell, I can prevent autoimmune disease, and blah, 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 and on and on and on. So what I'm trying to say here is don't restrict your vision about imaging. Imaging has tremendous capabilities in terms of posing and supporting experimental exploration. So the other aspect that I think is important now is to realize that because of the progress of computing and our ability to create better and better images, you can also study an organ over time. So one of the efforts that I was involved in at the NIH with Judy Rappaport was, what is the standard evolution of the human brain? What is it that happens in the human brain from birth all the way to adulthood? And so her project was to combine every single potential data point in a cohort of children from basically birth all the way. And you know, it's not so easy to get a study done like this, because you have to have the consent and the parents and so on. But the idea there is that, you know, by following this and by doing all of the both static and dynamic, including diffusion tensor imaging and so on, what you're doing really is you're creating a reference database. Essentially, this project, expanded, essentially creates a library of standards, right? And you can now say, do I need really a neurologist or a neuroradiologist who's going to look at the things? I think this patient is ahead of development or below development, or that's autism, or that's schizophrenia. Do you really think you can do this with the naked eye? No. Probably what is going to happen is that every one case is going to be templated against a standard rich database in a relational way. So you'd look at something and you say, oh, I'm surprised about the temporal lobe here, and you circle that, and it will search into the database. But the database doesn't have to be just normal. It will tell you it's 2 standard deviations of normal. And oh, by the way, it relates to another database where we've had autistic patients that are well characterized, or patients with Alzheimer's or whatever. So you can imagine now a world where it's almost like cloud diagnosing. It's not the usual diagnostic methods that we use in traditional radiology department. So you might be seeing a patient at Hopkins, you send that into the cloud and it says, well, your patient fits into this components of what we know human evolution of brains is, and so on, and you can do it all through life. So the normal phenotype becomes subcharacterized, and your diagnostic function is no longer what it used to be in the 20th century. It's a completely different function. So that's one idea that I think is going to, in my view, revolutionize pediatric imaging. The same thing is true here in the Alzheimer's disease neuroimaging initiative, and you know this has been probably the most productive collaborative efforts we've made over the past 10 years, because we discovered actually with these methods from Thompson at UCLA, with these sort of small representation of variation in different regions of the brain through this sort of M&M plots, that in fact you can actually identify early signs of Alzheimer's disease with relative precision. So this is the future in my view, without forgetting that we are going to also use imaging to change the way interventions are done. I think we know that already. I've always tell my surgeons, I hope there's no surgeon here, that 50 years from now there will be no surgery left. And they just basically don't believe that indeed this is possible. I believe that we will reduce the impact of surgical interventions by really going to combinations of scanner and computer advances and novel therapies. I think you know you have a pretty good group here. And epilepsy is a good example. The people at Yale have shown that by combining high field imaging, this is a patient that had unexplained epilepsy, no lesion was found. The lesion is actually here on a 1999 image, and this is a high field imaging, and you can see the lesion very well. So again, 60% of the foci of seizure can now be detected that wouldn't have been detected before these imaging techniques. So we can see that, in fact, once you see better, you can intervene directly. And once you see better, you can have other ideas about how to manage this disease. Combining the anatomic information with the electrical maps, it's happening as we speak, and it's really changing the way epilepsy is being treated. And then Brad Wood at NIH, but many other people have really worked on this. The concept of a molecular interventions where you combine a device, an imaging system, and a drug with targeting antibody or a ligand and the payload. I'm not telling you anything different. And here, the biophysics of these particles relative to the vascular bed, relative to the fenestrations in different organs, whether it's the liver versus the kidney, is amazing science, and especially in the 50 to 100 nanometer range. And it's not fully characterized, but we're getting there. I mean, it's very interesting that the tolerance of a particular organ to a size is amazing. If you're in the 70 nanometer range, it goes to the kidney. If you're in the 80, 90, it's predominantly picked in the liver. It's quite interesting physiology. And obviously, you will, with smart drugs, smart delivery, I don't have to tell you. So in my view, my conclusion is I've shared with you what I think are the major trends and challenges in fundamental biomedical research. I think I've tried to share with you what I see imaging being able to do in this century. And so the question is there a time for a new redefinition or a new vision? And the question fundamentally is imaging defined by its tools or its purposes? And what I mean by that is, are you someone who manipulates scanners and microscopes or whatever else? Or are you someone who uses imaging to solve problems? And that's a fundamental self-view that we as a specialty, as a field of science, have to resolve. Because too often, people define themselves by their tools. And it's almost like saying, if you have a hammer, are you the hammer specialist or are you a carpenter? So if you have a hammer, everything looks like a nail. And if you're a carpenter, you can change from a hammer to something else and adapt it. So we should really think about us having an integrated look about all non-invasive testing of live matter is really the field of application of imaging. Should we focus on specific and relevant applications of the imaging sciences to clinical radiology alone? And my view is, look at what we did in computing. I mean, this thing about creating relational image databases, concepts that relate to thousands of reference scans that can be used as an objective reference diagnostic database for your diagnostic radiology department, that would never come from the standard approach to clinical radiology. That will come from left field. We need to embrace left field. Well, should the discipline be a total science? I think, to me, the answer is obviously yes. I'm setting up the questions so that you can't come to the same conclusion I'm trying to get you to. But frankly, we need to have a unifying concept. So to me, what I'd like to say is, in the context of biomedical research, imaging should be defining itself as the core interdisciplinary science for extracting spatially and temporally resolved information. If it's not spatially resolved, I don't think we need to be there. And it has to be biological information. In other words, we're not just trying to read out what's there, but we're trying to actively extract biological information at all scales, angstrom, microns, in intact biological systems. So biomedical research, in my view, requires strong multidisciplinary effort in general, but more so in imaging. And this is where I think institutions need to understand that medical research today is not just the hospital and the medical school together, but it's a huge multidisciplinary environment with excellence in physical and biological sciences. And this is why, when I saw the BioX experience here, I thought this was the right thing to do. I think Stanford has been one of the most successful in getting the new grants that I created at NIH, the Innovator Award grants and the Pioneer Award grants with Carl Desserot, for example. I remember him because he couldn't get funded through the regular process, and he got a Pioneer Award. So I think Stanford is a good example of that, and this is why I thought that I would say this, because you're on the right track. I'm pleased to have been able to come here and share with you some of this vision, and also really, again, stress the fact that I don't think Stanford could do it without the leaders it has had, in particular Gary Glazer in radiology. Thank you very much. You want me to take questions? Okay. Questions? Yes.
Thanks for that excellent talk, Elias. One question about the limitations of imaging, in that if you go back to your systems biology concept and understanding networks, it seems that one key barrier is being able to interrogate in imaging large numbers of events simultaneously. And if you really are going to solve these complex disease states and are only able to observe 1, 2, 3 things at a time, can imaging in itself really succeed, or does it actually need partners that are?
Yeah, so your question almost implies a certain vision of imaging. See, to me, I think you need to really guide imaging to the point where spatially localized information becomes relevant to the disease process. So So it may be a combination where you would do an array, you would find 3, 4 targets that are relevant, and then you put it in a cell system, for example, and you say, gee, this prevents mitochondrial fusion. You can see that, locally see it. And then you can so interrogate back and forth between what I would say, base information. You can do silencing RNA experiment with imaging. People like Gary Rifkin have done this. So they went in, they repressed every gene in C. elegans, and they defined whether that would lead to an accumulation of fat or not. Well, you can see the kind of experiments you can do. Now, you're right. I mean, you're not going to be able to measure 100 factors with a full imaging experiment, but you can definitely enrich your understanding by having steps where imaging becomes a step of that. I don't know if I'm answering you, but the same is true in neurointerventions. I mean, one of the issues we have in neurotrophic factors is the lack of the ability to measure exactly what is happening. Well, with methods like optogenetics or other methods, at least in the experimental system, you can now. But you need imaging to do that. So, yes.
Paradox between
I used to think that imaging was costly, very costly, because of scanners and so on. But frankly, if you look at the real cost of imaging, it's almost like a very decreasing cost. I mean, I remember the scanners were $2 million. It's like computers. They were $2,000 in 1980. They're $2,000 today. And that means a huge decrease in cost. So to me, what I see the cost is not in the technology. That's what a lot of people say. And I don't agree with that. I think that if I could drive imaging to identify clearly patients at risk of Alzheimer's disease, and I could then use that to develop treatments that would just delay the onset of Alzheimer's disease by 5 years, I will reduce the cost of Alzheimer's by 50%. So I agree with you. I think it's a complex equation because the cost of technology is not the technology, it's the abuse of technology. Proper or inappropriate use of technology. So I think we suffer from inappropriate use of technology, imaging included, as opposed to technology itself being a problem. Now, if you hear about the cost of drugs and rare diseases and so on, I mean, you're talking about $200,000, $300,000. I mean, it's $100,000 for Avastin to lengthen life by 3 months. I tell you, I'll take an imaging thing that tells me if I can do that any day over that. So I agree that I didn't really want to cover the economics of that. Yes, Herb?
as you pointed out, there's been getting a better understanding. Speaking of
Yeah. So first of all, the notion, you're completely right, Herb. The question is, can you truly reverse Alzheimer's disease once established to the point of loss of function? And everybody that I know of says, I don't think. Once you've lost neuronal mass, it's like late-stage diabetes. Very hard to recover from that because the neurotrophic factors may not be there, the regrowth of neuron, the plasticity may not be there. So it's clear that the only hope is really to do it earlier. The second is when you really try to design an experimental approach to Alzheimer's disease, because the disease evolves so slowly, for you to do a trial in the late stages will take you 10 years, and it's too expensive to do that. So from the functional standpoint, you need to intervene early. From the practical experimental standpoint, you need to have markers. You need to find a way where you can really measure the impact. So at this point, what's really important is that now it's being used there are several trials that are ongoing right now, the results of which are not clear to me, and they're not published, but they will come. But for the first time, we have the ability to test many different approaches and have a readout not 10 years later, but 2 years later, 3 years later, and find out if anything that is being done changes the course of the disease. We can't alter and reverse the course of the disease, but we can change its course forward. And that's the hope and strategy for Alzheimer's disease. But nothing now has been, there's the antibody work against plaques, but the first result was surprising because the patients did have a clearance of the plaque, but they got worse. So clearly, again, it's one of those things where I don't believe we understand the fundamental pathology yet well enough to be good. Yes, I'm sorry. One more? Okay, one more.
I enjoy your talk, by the way.
Thank you. So we try some of it. So for example, the network biology, the biomapping, the understanding of translational medicine so that you can validate early. Because one of the problems in R&D has been that we've relied a lot on models, but they've not been predictive. So how do you change that? So we're trying to establish, for example, what we call phase 0 trials, where we interact with biomedical institutions to try to validate whether or not our mode of action is really happening, if indeed there is a readout that you can use in the human population. So weaving we call it translational medicine, but good practice of experimental medicine early in the discovery is very essential for what we do. In terms of imaging per se, I think we're going to rely on partners like we did with Sam or others in terms of answering questions. But I don't think we have significant imaging capabilities, animals and cells, but we're not going to be generators of imaging solutions. Thank you.
So Dr. Zerhouni will be available for a few minutes out in the lobby, and I'd like to encourage anybody who'd like to talk to him to do that. So you're talking to Sam at 11, and I thought we could just...
No, I'm not talking to Sam at 11. I have a phone call.
Oh.
You have an extra card?
I don't have a card.
Thanks, so it's excellent. I'll see you later.
I'll see you later, yeah.
Thank you so much.
Send you something from ancient period.
Okay.
That was great.
Thanks. Appreciate it.
Do you have a PC? How do you eject that?
Oh, it's a little different. You have to go to My Computer. This is funny. I just my fantastic talk. And you go to. This is my normal. This is his.
There's a dot in the middle.
I'm just
Just
ejecting
eject
his.
that.
Okay, it's ejecting.
Thanks for coming in.
Safe to remove hardware.
Are you aware that we have at Stanford the ultimate in high-resolution temporal and spatial imaging? It's an X-ray laser that's operating right here at SLAC.
Oh, SLAC, yeah. No, I didn't know that, actually. I know about SLAC, but I don't
We're
know.
doing imaging for nanocrystals,
Butts-Polly.
hopefully soon single biological proteins with single-shot pulses from this X-ray laser.
I'm in radiology. I'm in
Are you looking at molecular conformation and things?
Following things on a femtosecond time scale and angstrom spatial resolution. So those are
Good
the
stuff.
two ultimate scales that you talked about.
Yeah. That's good stuff,
You
actually.
should come see it. Are you here long enough to arrange a visit there?
No, I don't. No, you gotta go back tonight.
Oh, sorry.